Hello students and welcome to EPG Pathshala. Today we are going to discuss the various immune responses to bacterial infections from the paper Immunology. In this module we will start with a brief introduction where we will see what are the various mechanisms a body adopts to recognize the bacterial infections. Later on we will move on to the protective measures taken by the body to combat these bacterial infections. These mechanisms can be innate immune responses or the antibody-mediated humoral immune responses or the cell-mediated immune responses. Later on, we will move on to various mechanisms that bacteria utilizes to overcome this kind of protective measures taken by the host body. Lastly, we will discuss various immunopathological conditions arising because of overimmune responses taken by the body against these bacteria. We will end up this module with a brief summary of all these mechanisms. The immune response against bacteria depends on whether the bacterium is present extracellularly or intracellularly, that is the location of the bacteria. Also, it depends whether the pathogenicity is because of the invasion of the bacteria or simply by the release of toxins. In addition to that, the chemical structure of the bacterial cell wall, the degree of virulence and the inoculum size decides the type of immune response to be generated. The protective measures taken by body to combat these bacterial infections can be classified as innate immune responses, the antibody-mediated immune responses and the cell-mediated immune responses. The entry and establishment of the bacteria into the host body is first taken up by the first line of defense or simply the physical barriers of the body. It may include the complement system, the phagocytic activities, opsonization, etc. Besides these physical barriers, the body can adopt the antibody-mediated responses as well as cell-mediated immune responses depending on the location of the bacteria. On the other hand, bacteria also adopts various strategies to evade from the protective measures taken by the host. But at times, the protective measure taken by the body against the bacteria are not actually protective. The overimmune response cause harm or damage to the host body as well, and ultimately it leads to immunopathological conditions in the host. So students, let us start our module by trying to understand how a bacterial component is recognized by innate immune response. The immune mechanisms generated against pathogenic bacteria depends upon the chemistry of microbial surface, pathogenicity mechanism, the survival of bacteria in extracellular matrix or within the cell. The bacterial surface or cell wall differs in gram-positive, gram-negative, mycobacteria and spirochetes in terms of its chemical composition. Besides inner plasma membrane and peptidoglycan layer, gram-negative bacteria also have an outer lipid layer that contains lipopolysaccharides or LPS. The cationic proteins and complement proteins produce effective immune response against these outer lipid layer. Lysozymes and lysosomal enzymes also act against peptidoglycan layers. The gram-positive bacteria have lipotecoic acid that is recognized by innate immune response. The mycobacterial surface contains mycolic acid, glycolipids and liparobilirubin as potent immune recognizers. Fimbriae and flagellae in some other bacteria generate antibody response. In spirochetes, lipoproteins and murine are recognized by innate immune branch. Some bacteria cause disease because of release of toxin, for example, Corynebacterium diphtheriae, or attachment to host epithelial surface such as streptococcal sore throat. Antibodies can neutralize the function of such organisms and provide immunity. On the other hand, some bacteria cause disease because of release of toxin such as in case of Corynebacterium diphtheriae or attachment to host epithelial surface, for example, streptococcal sore throat. Antibodies can neutralize the function of such organisms and provide immunity. On the other hand, some bacteria are not toxic as such, but due to their invasion and accumulation in cells and tissues or from immunopathology disease is caused, such as in case of lepromatous leprosy. Cell-mediated immune response is required to achieve immunity in such cases. But most of the bacteria fall in between the above two extremes, that is, they cause local toxicity and local invasiveness. For such bacteria, both a humoral as well as the cell-mediated immune response is required to achieve the immunity. Let us see what are the major mechanisms that operate in human body to combat bacterial infection. It can be neutralization of bacterial toxins or enzymes by host antibody, lysis of bacteria by complement and antibodies, bacterial opsonization by complement leading to phagocytosis or destruction, intracellular destruction of bacteria by activated macrophages, or direct killing of bacteria by natural killer cells and cytotoxic cells. You can see all these mechanisms which have been summarized in this diagram. Let us see what are the innate immune response against bacteria. The entry and establishment of pathogenic bacteria is inhibited by simple barriers which constitute the first line of defense. These non-specific innate immune barriers include impermeability of skin, fatty acid production by skin, ciliary movements on trachea, flushing in urinary tract, pH in stomach and vagina, lysozymes, etc. Various commensals limit the invasion of bacteria. The endogenous microbiota in our gut competes with pathogenic exogenous bacteria for nutrients by stimulating immune response and by producing antibacterial proteins such as colicins and microcins. Since these resident bacteria are also not completely immune inert, the host immune system restricts them by minimizing their contact with epithelial cells, production of antimicrobial peptides and secretion of IgA. Still, if subcommensals break these barriers, dendritic cells and cell-mediated immune response protect the host against them. The soluble molecules and cell surface receptors. Before the B and T cell mediate response, various soluble molecules and cell surface receptors recognize the distinct molecular patterns on microbial surface called as pathogen associated molecular patterns or PAMP, or they are also called as microbe associated molecular patterns or MAMP. The molecules that recognize them are called as pattern recognition receptors or PRRs. The PAMPs include carbohydrate, lipoproteins and lipopolysaccharides present on the microbial surface. Many non-pathogenic cocci are removed from the tissues as a consequence of these pathways without any adaptive immune response. The products of damaged cells are termed as danger associated molecular patterns or DAMPs. The PRRs are categorized in three categories as intracytosolic recognition molecules, cell surface and endocytic vesicle receptors, secretory molecules in serum and body fluids. Let us discuss them briefly. The intracytoplasmic recognition molecules or cytosolic receptors recognize the intracellular pathogens, that is bacteria and virus. For bacterial recognition, the nucleotide binding and oligomerization domain like receptors, for example NLR, are involved which recognize bacterial peptidoglycans. On the other hand, for viral recognition, retinoic acid inducible gene 1, that is RIG1, like helicases, that is RLHS, are there which recognize double stranded RNA. The NLRs are a part of multiprotein complex inflammasome which cause pyroptosis, that is cell death due to inflammation of infected cells. The node 1 and node 2 recognize bacterial peptidoglycans which activate NF-kappa B and MAP kinase pathway through RICK, which further leads to activation of caspases and production of IL-8 and IL-1 beta. Moving on to other category, that is cell surface and endocytic vesicle receptors. All these PRRs help phagocytes to recognize the pathogens. The recognition of pathogens by phagocytes can be indirect or direct. The indirect recognition involves deposition of serum derived molecules on pathogen surface followed by their binding to phagocyte receptors. Complement proteins such as C3b, collectins, ficolins and pentraxins are some of the opsonin molecules that are involved in such indirect recognition. In case of direct recognition, the receptors present on the phagocyte surface recognize the PAMs, which include toll-like receptors, carbohydrate receptors and scavenger receptors. In this table, you can see various toll-like receptors along with their PAMs and the associated pathogens. The toll-like receptors are named as TLR 1, 2, 3, 4, 5, 6, 7, 8 and 9. Moving on to the scavenger receptors. Scavenger receptors of macrophages are involved in uptake of apoptotic bodies, modified lipoproteins, polyanionic ligands and some bacteria. Class A scavenger receptors include SRA1, SRA2, MARCO and CD163, whereas CD36B and SR plus B1 are classified as class B scavenger receptors. The next category, that is mannose binding C-type lectins. For example, the DC sign are carbohydrate receptors expressed on some macrophages. It has been implicated in interaction between APCs and T cells and in microbial recognition. It modulates TLR signaling to promote expression of cytokines IL-10 and IL-8. The secreted molecules in serum and body fluids, the acute phase proteins, are some of the secreted molecules in blood whose levels increase during infection. For example, C-reactive protein is increased approximately 1000 times during inflammation or infection. Besides this, a variety of molecules are secreted from macrophages, for example, the lysozymes, complement C3b, reactive oxygen and nitrogen species, cytokines, etc. Moving further, we do have lymphocyte independent effector mechanisms which are operated in the body to combat bacterial infection. The first among them is complement mediated effector response, that is direct lysis of bacteria whose lipid bilayer is susceptible to membrane attack complex, opsonization of bacteria and further in an interaction with phagocytes by attachment of cleaved products of complement C3. C5a attracts and activates neutrophils which cause degranulation of mast cells, leading to release of histamines and leukotrienes that cause vascular permeability. The next category is cytokine mediated effector response, that is increase in adhesive properties of vascular endothelium that facilitates passage of phagocytes to inflamed tissue sites, is called by cytokines such as TNF and IL-1. Cytokines along with chemokines CCL2, CCL3, CXCL8 help in recruitment of leukocytes. Acute phase response is initiated by cytokines IL-1, IL-6 and TNF which enhance complement production. IL-12 and IL-18 stimulate NK cells which release IFN gamma that further activates macrophages. Category is pathogen recognition based regulation of lymphocyte mediated response. Besides generation of innate immune response, regulation of development of lymphocyte mediated immune response is also carried out by pathogen recognition based on signaling mechanisms. Some of these mechanisms are augmentation of antigen presenting abilities of immature dendritic cells or the binding of bacterial components with pattern recognition receptors. The phagocyte mediated bacterial killing. Most of the extracellular and intracellular bacteria are killed by phagocytic activity of macrophages and the neutrophils. The process is carried out in following steps. The first step is chemotaxis of phagocytes to the bacterial components. The phagocytes are attached to bacterial pathogens by bacterial component F-methyl-leucine phenylalanine, the cytokines and chemokines and the complement product C5a. The type of pathogen and time since infection determines the phagocytic cellular composition in inflammatory response. For example, the acute inflammation with Streptococcus pyogenes causes neutrophils rich lesions, whereas the chronic inflammation with Mycobacterium tuberculosis results in macrophage rich granuloma. Salmonella infection leads to mixed population lesions. The next step is binding of phagocytes with pathogen. The phagocytes may bind directly to the bacteria by pattern recognition molecules or indirectly through complement receptors. Uptake of pathogen by phagocyte is the next step. After chemotaxis and binding, the uptake of pathogen takes place, which is enhanced by macrophage activating cytokines. For example, the granulocyte macrophage colony stimulating factor GM-CSF. Lastly, the killing of bacteria by phagocytes takes place. Once internalized, the pathogen is bound by host membrane phagosome, which confines it to deliver antimicrobial molecules at high concentration and avoid damage to the host. The killing mechanism may be acidification of phagosomes, generation of reactive oxygen intermediates, promotion of influx of potassium ions in the phagosome, activation of microbicidal protease by fusion with lysozymes, or the creation of nitric oxide and peroxynitrite from guanidinonitrogen or L-arginine. Yet another mechanism is the production of antimicrobial peptides. Besides the all above stated mechanisms, various cationic proteins possess antibiotic-like properties. For example, the defensins. The phagocytic cells such as neutrophils contain antimicrobial peptides called as defenses. These defenses are 30 to 33 amino acid long with cysteine and arginine rich cationic peptide moieties. They help in lysis of bacteria by integrating within their lipid membranes and disrupting their cellular integrity. They are very important in killing bacteria such as Pseudomonas, E. coli and Staphylococcus. Defensins have been classified as alpha defensins, beta defenses, theta defenses, etc. The example include HBD14 and HNPs. Another antimicrobial peptides are cathelicidins. These are another class of antimicrobial peptides found in neutrophils, epithelial cells and phagocytic cells. There are around 12 to 100 amino acid long peptides which help in bacterial membrane disintegration. In humans, LL37, HCAP18 are prominent cathelicidin antimicrobial peptides. Protegrins is yet another class of antimicrobial peptides which are composed of around 16 to 18 amino acids, chiefly the arginine and cysteine. They have been shown to have microbicidal properties against Candida albicans, Listeria monocystogenes, Neisseria gonorrhoeae, etc. The mechanism of action of protegrins is similar to other antimicrobial peptides, that is, disruption of membrane of pathogens. Cathepsins G and azurocidin are cationic peptides with different pH optima and related to elastase, but they act against gram-negative bacteria unlike the enzyme activity. The neutrophil extracellular traps release the neutrophil extracellular trap as mechanism to kill the extracellular bacteria, which involves release of antimicrobial peptides along with the histones and chromatin from activated neutrophils to kill the bacteria as well as to avoid the spread of infection. The other strategies may involve restriction of nutrients and iron to intracellular bacteria, etc. Students, now let us move on to the evasion strategies which various pathogenic bacteria adopt to avoid the phagocyte mediated killing. The pathogenic bacteria have evolved a number of protective strategies to evade themselves from phagocyte mediated killing, such as secretion of toxins to inhibit chemotaxis, inhibition of attachment of phagocyte by capsule or outer coat formation as in case of Streptococcus, release of factors that block the signaling mechanisms for killing the bacteria, secretion of catalase to break down thereby inhibiting RNA production, the free radical scavenging activity by release of some phenolic compounds from the outer coat, release of compound such as lipoarabinomannan from Mycobacterium is another strategy which blocks the ability of macrophages to respond to IFN gamma and thus inhibit their activation. Impairment of antigen presenting function is yet another strategy employed by Salmonella and Chlamydia. Shigella and Listeria avoid the phagosome and multiply in cytosol. Some bacteria induce apoptosis and necrosis to kill the phagocytes itself. Let's move on to humoral immune responses taken by body against bacteria. The antibacterial defense by antibodies and or B lymphocytes, such as the neutralization of toxins by blocking the attachment of binding portion of the molecule to its target cells. It can also block locally acting toxins or extracellular matrix degrading enzymes which act as spreading factors. It can interfere with the bacterial motility by binding to the flagella. Secretory IgA prevents bacterial binding to epithelial cells. Some antibodies to the bacterial surface can block functional requirements of these bacteria such as nutrient uptake. Antibodies provide more efficient targeting of complement to non-toxigenic bacteria. Specific high affinity IgG antibodies play important role in antitoxin response. Antibodies against fimbriae, lipotecoic acid and some capsules hinder the attachment of bacterial pathogens to host surface. Antibodies to AM proteins and capsules provide opsonization via Fc and C3 receptors. Antibodies may also neutralize immunorepellents. Students, let's have a look on the evasion strategies which bacteria adopt to avoid the effect of antibodies. The pathogenic bacteria have evolved various strategies to avoid the antibodies. Neisseria gonorrhoeae, the pathogenic bacteria that cause a sexually transmitted disease gonorrhea, adopts a number of mechanisms to safeguard itself, such as it does not evoke a large antibody response, for which the mechanism is still unknown. It generates the antibodies that block the function of killing or damaging antibodies. It also releases blebs of membrane that absorb and deplete local antibody response. It also alters its antigenic composition by sialylation of its lipopolysaccharide, undergoing phase variation of surface antigen expression and generating variants of pilin by homologous recombination. Besides this, Neisseria also impairs T cell activation as well. Adopt complement mediated evasion strategies as well. For example, the outer capsule formation that prevents binding and activation of complement factors. In case where complement C3b has been fixed on bacterial membrane, the outer surface configures itself in a way that complement receptors present on the phagocytes cannot have access to it. Thus, complement mediated phagocytic activity is inhibited. The terminal pathway of complement leads to binding of membrane attack complex to bacterial surface that lyses it. Some bacteria express surface structures that divert the attachment of this lytic complex. The membrane possesses certain enzymes which can degrade the fixed complement proteins or shed them. The outer membrane may also configure itself such that the insertion of lytic complex is resisted. The bacteria may also secrete some decoy molecules that bind the complement proteins and avoid their fixation on bacterial surface. Some bacteria contain long side chains, that is O antigens on their LPS. These O antigens fix C3b on distant sites, disallowing them to bind to the lipid bilayer. Factor H and I inactivate the attached C3b. The capsules which are rich in sialic acid promotes the interaction with these factors and thus inhibit the complement system. Moving on to the last category, that is cell mediated immune response against the bacteria. The infected cells can be killed by cytotoxic T lymphocytes. The intracellular bacteria pathogen can be classified as cytosolic or phagosomal. The cytosolic pathogens are processed and presented via MHC class I molecules to naive CD8 positive T cells. The phagosomal pathogens induce CD8 positive T cells via cross priming, in which infected and apoptotic cells release antigenic fragments which are taken up by the dendritic cells. The effector CD8 positive T cells or cytotoxic T lymphocytes provide immunity by releasing pro-inflammatory cytokines, macrophage activating cytokines, perforins or granulysin release etc., which lead to killing of pathogen. Here you can see the antibacterial response of T lymphocytes against cytosolic and phagosomal bacterial pathogens, which is leading to bacterial lysis. Students, now let us move on to various immunopathological reactions which are induced by overimmune response to bacteria. The immune response mounted against invading bacteria have potential to cause damage to host as well, and thus it leads to various immunopathological reactions in the host. Some of the examples are the endotoxin shock or sepsis. A sudden and massive cytokine release in response to bacterial subunits leads to pathological condition which is called as endotoxin shock. Fever, circulatory system collapse, hemorrhagic necrosis and hypothermia are the common features of such an endotoxin shock. It further leads to multiple organ dysfunction syndrome or MODS and ultimately leads to death. The initial response is called as systemic inflammatory response syndrome. Individuals who survive from it often undergo more dangerous compensatory anti-inflammatory response syndrome, in which the individual becomes more susceptible to secondary infections due to compensatory anti-inflammatory response. Another example of immunopathology is T cell stimulation by superantigens. Some bacterial components are called as superantigens. These superantigens bind directly to variable region of beta chains of antigen receptors on T cells and cross-link them to MHCs present on APCs, usually outside the normal antigen binding groove. Due to this, a massive release of cytokines takes place. The toxic shock syndrome due to staphylococcal toxin is an example of such superantigen induced T cell activation. In this diagram, you can see the difference between a normal and superantigen mediated T cell stimulation. So, students, let us finally summarize what we have all learned in this module. The immune mechanisms generated against bacterial infections depends on various factors. These factors may be the first one, the surface, the chemistry of the surface of the bacteria which is invading the host organism. It can also depend on the degree of virulence, on the mechanism of pathogenicity which is being adopted by the microbe, whether it is going to be invasive or simply releasing some local toxins. Additionally, it also depends on the location of survival of the bacteria, whether it is extracellular or residing intracellularly. Later on, we have seen various mechanisms that operate within the body to combat these bacterial infections, generally classifying them as the first can be simple neutralization of toxins. It can be the lysis of bacteria by complement system or the antibodies. It can be a direct lysis of the bacteria by cytotoxic T cells or the natural killer cells, the opsonization, etc. Before B cells and T cells mediate the response, various soluble factors and the surface receptors recognize the patterns which are presented on the microbial surface. These patterns are called as PAMPs or pathogen associated molecular patterns. The receptors which recognize them are called as PRR or pattern recognition receptors, which can further categorize in the intracytoplasmic receptors, the soluble factor receptors or maybe the surface receptors. Besides these, there can be lymphocyte independent as well as lymphocyte dependent mechanisms which are operated in the body to combat these bacterial infections. The lymphocyte independent ones can be the complement mediated, cytokine mediated or the antimicrobial mediated mechanisms. Additionally, when we go for lymphocyte dependent mechanisms, it can be further categorized whether the B lymphocyte or antibody mediated or the T cell mediated. The B cell mediated lysis along with the complement mediated lysis is generally adopted for extracellular pathogens, whereas the direct killing by T cytotoxic cells involve the killing of bacteria which are intracellular in nature. Besides all these mechanisms which body adopt, mechanisms have been devised by the bacteria to evade themselves from all these mechanisms. Additionally, the damage is caused not only to the host bacteria, but to the host as well. The examples include the endotoxin shock or the sepsis or T cell stimulation by superantigens. Both of these arise because of overstimulation of the immune response against these bacterial infections. Thank you.